Hello everyone, welcome back to my channel. So today we are moving on to the next topic from the chapter Biotechnology Principles and Processes. Okay, so I hope you remember the topic what we have discussed in the last class. So we were discussing about the first topic, the introductory part of biotechnology, and after that I told you about how to construct or like how the Stanley Cohen and Herbert Boyer constructed the recombinant DNA. Okay, so those topics that we were already discussed in the last class. Now we are moving on to the topic like tools of recombinant DNA technology. As I have already told you that for making recombinant DNA, we have to use many tools. Okay, we need a molecular scissor for cutting the DNA, and we need a molecular glue for joining the DNA, and as well as we need DNA polymerases, we need a host, so and so. So we are moving on to each and every tools which are required for making a recombinant DNA. So the first tool of making a recombinant DNA, what we are going to discuss is actually restriction endonucleases, or we can say that molecular scissor. Okay, molecular scissor for cutting DNA. Now, see, in restriction endonucleases, first we have to like discuss about the discovery or history of this restriction endonucleases. See, in the year we can say that 1963, actually two enzymes which are restricting the growth of bacteriophages were identified in E. coli. Okay, two enzymes which are restricting the growth of bacteriophage. So these enzymes they are restricting the growth. They are not allowing the growth of bacteriophages which were discovered from E. coli. So the enzymes were discovered from E. coli. Okay, so now one of the enzymes they will add methyl group to the DNA. So one of the enzymes they will add methyl group to the DNA. So they will add a methyl to the their own DNA, so they can easily recognize the foreign DNA from their own DNA. Okay, so that is actually the first like enzyme, and the second enzyme it will cut the DNA. So later was they termed as actually restriction endonucleases. Okay, restriction endonucleases. Now, see the first restriction endonucleases which were discovered is actually HIND2. So HIND2 is actually the first restriction endonucleases. Very important. HIND2 is the first restriction endonucleases. Okay, so it is actually discovered from Haemophilus influenzae. Okay, it is discovered from the Haemophilus influenzae. Now, see this restriction endonucleases, as we have already studied that they will inspect the DNA. So once they will recognize the recognition site, or once they will identify the recognition site, or we will call it as palindromic site. Palindromic site is actually a six-letter word which will be same when we will read forward and backward. That is actually the palindromic site. So when they will identify the palindromic site, there they will act. Okay, that is actually the restriction endonucleases. Now, see approximately, see this is actually the first restriction endonucleases. Approximately 900 restriction endonucleases are actually identified from 230 strains of bacteria. Okay, so from 230 strains of bacteria, approximately 900 like restriction endonucleases are actually identified. Okay, that you have to keep in your mind. Now, see the restriction endonucleases, what we need to study, that is actually eco R1. Eco R1, that is a restriction endonucleases what we want to study, or it is actually given in the NCERT. Now, so we were discussing about restriction endonuclease. I told you that it is actually, just imagine this is actually DNA. For cutting this DNA, see, for making this fragment as like this, for making fragment, we required a molecular scissor. That is actually the restriction endonucleases. Okay. Now, naming of restriction endonucleases. See, naming of restriction endonucleases, first we can start from the eco R1 itself. Okay, see here eco R1. See that here it indicating actually it is isolated from Escherichia coli. So the bacteria Escherichia coli. So the full name is actually Escherichia coli RY13. That is the bacteria. Escherichia coli RY13. Now here in Escherichia coli, see the E, the first letter E that representing the genus name of the bacteria. So Escherichia. E is actually the genus name, Escherichia. Okay. Now the second two letter that representing the species name of the bacteria, that is actually coli. Okay, C-O-L-I. Coli. Escherichia coli. Now, R is actually the strain of bacteria. Okay, the type of bacteria. I told you RY13. Okay, R is the strain of bacteria. Now, 1, the Roman number that indicating the order in which this enzyme isolated from the strain of bacteria. Okay, this strain of bacteria. Okay, so this enzyme isolated from this strain of bacteria, that is actually the Roman number. Okay, so that's all about actually the naming. So in the naming, the first letter representing the genus name of the bacteria, second two letter representing the species name, the R representing the like a strain of bacteria, Roman number indicating the order in which this bacteria isolated from the strain of bacteria. Okay. Now, in this also, simply we can elaborate one. H indicating, I told you that it is actually isolated from the Haemophilus. So, Haemophilus. So, second two letter IN, influenzae. D is actually the strain of bacteria. The Roman number indicating the order in which this enzyme isolated from the same like a strain of bacteria. Okay, so that is actually the naming of restriction endonucleases. Now, see this restriction enzymes, they belongs to a class of enzyme or a class, we will call it as actually nucleases. So restriction enzymes, they belongs to a class of enzymes called nucleases. So there are two type of nucleases. There are two type of nucleases. First one is actually the exonucleases. So exonuclease. Second one is actually the endonucleases. So first one, I told you that exonucleases. Second one is actually the endonucleases. Okay. So in case of exonucleases, we can say that see in exonuclease, it will remove the nucleotide from one end of DNA. Exonucleases remove the nucleotide from one end of DNA. Just imagine this is the DNA. Okay, this is the DNA and this is actually one end of DNA. So a DNA is made up of nucleotide. We already know it. So this enzyme, it will remove the nucleotide from one end of DNA. So that is actually called exonucleases. So endonucleases, already we discussed that it will cut the DNA. Okay, once if it will identify the palindromic sequences, it will cut the DNA. That is actually endonucleases. Okay, so here exonucleases, endonucleases. So already we discussed about the restriction endonucleases. Now we are moving on to the action of mechanism of restriction endonucleases. So as I have already told you that palindromic sequences. So we were discussed that each restriction endonucleases have a palindromic sequences. So once if they identify the palindromic sequences, they will cut the double-stranded DNA. So that is actually the action of restriction endonucleases. Now here, you just keep in your mind, see, actually this is actually the palindromic sequences of eco R1. So the eco R1, in eco R1, the palindromic sequence is actually GAATTC. Okay, GAATTC is the palindromic sequences of EcoR1. Now here GAATTC, GAATTC, so that you keep in your mind. So already we discussed a palindromic sequence is actually a six base pair word. So which will be same when we will read forward and backward. So that you have to keep in your mind. Now see here, you just keep in your mind. So this is actually the vector DNA. So this is the vector DNA and this is actually the foreign DNA. So as I have already told you that, see in case of vector DNA and foreign DNA, see just in the first video, I told you how to construct a recombinant DNA. So for constructing a recombinant DNA, there we have to use a vector DNA and a small fragment of DNA. So that I have already told you. And we should have to join the vector and the fragment DNA. Okay. Now here, so the very important point, if we are using a restriction endonucleases for cutting a fragment DNA, the same restriction endonucleases we have to use for cutting vector DNA. Have you got it? If we are using a like restriction endonucleases for cutting a fragment DNA or for cutting a foreign DNA, so the same restriction endonucleases we have to use for cutting vector DNA, because each of the like restriction endonucleases, the palindromic sequences are different. So if we are using different restriction endonucleases for cutting like foreign DNA and vector DNA, it will make different fragments. So it will be very difficult to, or it will be complicated to join or make a recombinant DNA. Okay, so that is the reason why we have to use the same restriction endonucleases for foreign DNA as well as in the vector DNA. So just keep in your mind, this is the vector DNA, this is the foreign DNA. Now in the vector DNA, actually here we are going to cut the vector DNA by using EcoR. Okay. So in the vector DNA, so what is the palindromic sequences of EcoR1? That is actually GAATTC. Okay, here also GAATTC. Okay, I told you palindromic sequence, when we will read forward and backward, it will be same. Okay, now so once it will actually, when we will add an restriction in the nucleases, it will inspect the length of DNA, and then once it will identify the specific palindromic sequences, they will cut the palindromic sequences. Okay, so they will cut the sugar phosphate backbone, that you keep in your mind. Okay, now so they will cut the palindromic sequences. So in palindromic sequences, see the restriction in the nucleases, it will cut a little away from the center and between same bases of opposite strand. Okay, a little away from the center but between same bases of two opposite strand. That also you have to keep in your mind. And when they will cut, we will get, see this is actually the way how they will cut, and after that we will get a single stranded portion. So these single stranded portion, we will call it as actually sticky end. Okay, so this single stranded portion is actually called a sticky end. Okay, now here, what is actually sticky end? Sticky end or cohesive end or staggered end? We can say in different names, sticky end or cohesive end or staggered end. So when we will cut the vector DNA, we will get a sticky end, and when we will cut the foreign DNA also, we will get a sticky end. Okay, now the sticky end, see, they are complementary to each other. So you can look at here, adenine pair with the thymine. This we have already studied in the chapter molecular basis of inheritance. Adenine pair with the thymine with the 2 hydrogen bond, guanine pair with the cytosine with the 3 hydrogen bond. This is in case of DNA. So in case of RNA, adenine pair with the uracil with the 2 hydrogen bond, guanine pair with the cytosine with the 3 hydrogen bond. Okay. So as I have already told you that, see here EcoR1, it will cut the DNA a little away from the center, but between the two bases of opposite strands, that you have to keep in your mind. and here it will leave a sticky end or single stranded portion or a overhanging structure. Now these two are complementary. So you can look at here. Here it is adenine, it is thymine. Okay, and adenine, thymine, thymine, adenine, thymine, adenine. So they will make a double stranded. Okay, so here the sticky end it will join together by using an enzyme which is called DNA ligases. So this is actually the action of like restriction endonucleases. Okay, so that you have to keep in your mind.